Previously, we've taken a look at the innate immune response, and we've taken a look at what types of cells are involved in the innate immune response and what those different cells can do. Macrophages and neutrophils are examples of cells that are considered to be part of the innate immune response. What we're going to take a look at here is the adaptive immune response, and B cells and T cells are examples of cells that are part of the adaptive immune response. The difference between innate immunity and adaptive immunity, just like the name implies, basically is that innate immune cells, these are cells whose ability to take care of pathogens are encoded for by genes that tend not to change over time. On the other hand, when you consider adaptive immune cells, the reason why they're called adaptive is because the genes that encode for their ability to take care of pathogens, those do tend to change. So for example, whenever a B cell encounters an antigen, it can end up changing itself so that it's better able to deal with that pathogen the longer that it sees it. So B and T cells, which are cells again of the adaptive immune response, they basically have an ability to become fine-tuned so that they are better able to deal with a pathogen over time. So let's sum that up here. Adaptive immune cells, the reason why they're called adaptive is because they can change after contacting pathogen to better deal with that pathogen. And basically the reason why they're able to change their characteristics is because they possess genes that are more prone to change. And we'll see a little bit later that examples of ways that the genes of these types of cells can change is through hypermutation and something else called gene recombination. So again, we've already taken a look at the cells of the innate immune response, and what we're going to do here is focus in on the cells of the adaptive immune response. We should mention that there's actually a lot of crosstalk between these two arms of the immune system. So for example, macrophages oftentimes work together with T lymphocytes or T cells in order to try to get rid of a pathogen. Right? So even though they're considered to be two separate arms of the immune system, they're not really separate. They work together. And it turns out that there are some cells that are actually hard to categorize. So there's some cells that dabble in both the innate and the adaptive arms of the immune response, and it's hard to say that it's strictly one or the other, because again, there's a lot of crosstalk between the two. So before we take a look at how cells of the adaptive immune response works, we'll first say something about leukocytes or white blood cells in general. And so all four of these cells that you see up on the screen right now, those are all examples of leukocytes or white blood cells. And again, whenever you say white blood cell, you're basically talking about a cell that functions in order to protect us against infection. And so it turns out that all white blood cells come from a type of stem cell called a hematopoietic stem cell. Hema means blood. Poietic basically means that it's pliable, so it could become multiple different types of cells. So whenever you're talking about a hematopoietic stem cell, you're talking about a stem cell, or you're talking about a cell that could potentially become any type of cell that you find in the blood. And so again, it's these hematopoietic stem cells that give rise to all of the cells that you find circulating in the blood. Hematopoietic stem cells can receive one of two types of signals. So there's some signals that tell these hematopoietic stem cells to become something called a common myeloid progenitor. And there's other signals that tell these hematopoietic stem cells to become a type of cell called a common lymphoid progenitor. And so it's the common myeloid progenitor then that ends up giving rise to all of these cells that you see here. So erythrocytes or red blood cells come from this type of cell. All of these cells that we've already talked about as being part of the innate immune response, they also come from this type of cell. On the other hand, the cells that you tend to think of as being part of the adaptive immune response, so in other words, things like B cells, T cells, plasma cells, those come from this type of progenitor cell called a common lymphoid progenitor. Right? So, but the bottom line here is that all of these cells that you find in the blood, all of these white blood cells, including actually red blood cells, come from a type of cell called a hematopoietic stem cell. And where you find these hematopoietic stem cells is in the bone marrow. And bone marrow is basically material, the substance that you find inside the bones. There's two types of bone marrow. There's yellow bone marrow, which basically contains a lot of fat, and then there's red bone marrow. Red bone marrow is the type that we're going to be concerned with here. And that's because red bone marrow is the type of bone marrow that contains these hematopoietic stem cells that give rise to all of the white blood cells that we're going to be talking about. And so where you find red bone marrow, it's actually shown in this picture here. All of the parts that are highlighted in red is red bone marrow, and so you find it in the sternum, in the bones that you find in the shoulders, also in the hips, and then the tops of the legs. And so basically here is where all of the white blood cells end up being produced. And after they're produced in the red bone marrow, they end up populating the entire body. And so that's what we'll take a look at next. How do the cells that are made in the bone marrow end up all throughout the body? And so like we mentioned, all of those white blood cells, they're produced in the bone marrow. From there, they actually enter the circulatory system. So they enter the blood pathways. But it turns out that it doesn't stop there. So the white blood cells aren't confined to the circulatory system. They can actually leave the circulatory system towards certain tissues. So that comes in handy whenever you have something infecting certain tissues, that you have the white blood cells able to leave the circulatory system and rush towards the tissue where the infection is occurring. Not only that, but the white blood cells that end up in the circulatory system can also enter something called the lymphatic system. And so we'll actually say a little bit more about the lymphatic system here in a minute. In other words, what does the lymphatic system do? What's its function? But the point we're trying to make with this slide is that white blood cells, leukocytes, they're found all throughout the body, not just the circulatory system. And it's actually a loop, right? So the white blood cells don't just leave the circulatory system. The white blood cells end up coming back to the circulatory system. So it turns out that the white blood cells can move between the tissue and the lymphatic system as well. And those white blood cells can go from the lymphatic system back into the circulatory system. So again, the white blood cells, or the leukocytes, they're found all throughout the body. And so we mentioned that one of the places where you find white blood cells is this thing called the lymphatic system. So what we'll take a look at next is, well, what is the lymphatic system, and why is it useful to have white blood cells populating the lymphatic system? So we'll take this picture and blow it up even bigger. Basically, I see the lymphatic system as the arena in which our white blood cells can do battle with any pathogens that are going to try to do harm to us. So basically, all of the white blood cells that end up being produced in the bone marrow, a lot of them make their way into the lymphatic system. We have cells that are able to take pathogens and then deliver them to the lymphatic system. So basically, this is where the good guys and the bad guys can come together, where hopefully the good guys, in other words our white blood cells, will win, where they can defeat the pathogens. And so basically the lymphatic system, here we'll describe it here, it's a network of vessels and organs that, like I just said, brings leukocytes together with pathogens. And so all of the green here in this picture represents the lymphatic vessels. It turns out that there's a fluid that runs through all of these lymphatic vessels. The name of that fluid is lymph. And so it's useful to have this fluid because as this lymph runs through the lymphatic vessels, that's what allows the pathogens in our white blood cells to come together. It turns out that there are several organs that all of these lymphatic vessels basically connect to one another. And you can categorize those lymphoid organs as being one of two types. And so to make it easier to see the organs that are part of the lymphatic system, we'll show you a different picture that doesn't show the vessels, but just shows the organs of the lymphatic system. There are several organs that you see here, and again, you can categorize them all as being one of two types. So one general type of lymphoid organ are these things called primary lymphoid organs, and there's actually just two lymphoid organs that you can say are primary lymphoid organs, and that's the bone marrow and the thymus. The reason why these are called primary lymphoid organs is because here are where the white blood cells are produced, and they end up maturing here. Right, so primary lymphoid organs are where all the leukocytes or white blood cells are made, and where many of them mature. It turns out that, so we mentioned at the very beginning that there's a type of adaptive immune cell called a B lymphocyte or a B cell. And it turns out that those are produced and mature in the bone marrow. There's another type of adaptive immune cell called a T cell. And it turns out that the production of T cells begins in the bone marrow, but then they end up migrating over to the thymus, and it's in the thymus where the T cells end up maturing. So we'll mention that here. All right, so again, all of these are considered to be organs of the lymphatic system. There are two, the thymus and the bone marrow, that are considered to be primary lymphoid organs. Primary meaning first, because here are where the white blood cells are first made. All of these other organs you see in this picture are considered to be secondary lymphoid organs. And it's at the secondary lymphoid organs where the battle occurs, right? So all of the lymph that goes running through the lymphatic system, they bring all of the pathogens and then all of the white blood cells in the lymphatic system over to these secondary lymphoid organs where the two can come together, and where hopefully again the white blood cells can prevail in getting rid of the bad guys. So in writing here, the secondary lymphoid organs, that's where pathogens are brought together with leukocytes or white blood cells, and once those leukocytes see the pathogen, that's when they become activated. And so, like you see in this picture, examples of organs that are considered to be secondary lymphoid organs where white blood cells get together and attack the pathogens include things like the tonsils and the adenoids, lymph nodes, and you actually find bundles of lymph nodes in various places, like in the neck and then in the armpits. There are these secondary lymphoid organs called Peyer's patches that are right next to the gut. So a lot of the pathogens that enter in through the digestive tract, the way that our immune system takes care of them is because they're taken over to these things called Peyer's patches, which are right next to the gut, and that's where our immune cells are able to contact them and take care of them. It turns out that there's a subcategory of secondary lymphoid organs. So again, all of the organs here, besides the thymus and the bone marrow, are considered to be secondary lymphoid organs. A subcategory of lymphoid organs are those that are really closely associated with mucosal surfaces. So in other words, there are secondary lymphoid organs and tissue that are closely associated with the breast, respiratory tract, genitourinary tract, and gastrointestinal tract. So taken all together, when you're talking about lymphoid organs that are really closely associated with these mucosal areas, altogether those are called mucosa-associated lymphoid tissue, or MALT. Right, so just a piece of terminology that you might find useful because it's used in your textbook in some places. But the point here is that the lymphatic system, it's the arena where the white blood cells do battle with any pathogens that try to infect us. It's where those white blood cells become activated as a result of them contacting the pathogens that try to infect us. So then going back to this picture here, we're saying that leukocytes, they're found all throughout the body. They start in the bone marrow, then go to the blood, then they end up in the tissues and the lymphatic system. What we'll do next is say a little bit more about how white blood cells get from the circulatory system over to the lymphatic system. And so say that here is a capillary that is a part of the circulatory system. Through this capillary runs the blood and the white blood cells that it contains. It turns out that the contents of this capillary aren't necessarily stuck in this capillary, so it's possible for things to seep out of the capillary. So for example, plasma can seep out of the capillary, and plasma is basically just the liquid portion of the blood, and it can contain antibodies, for example. And the reason why plasma might want to seep out of the blood is because it can then bathe the surrounding tissue. And one of the reasons why it might be a good thing to bathe tissue with plasma, again, is because plasma contains these things called antibodies, which could end up protecting this tissue against certain pathogens. And just a little tidbit, whenever plasma leaves the capillary in order to kind of bathe the body's tissues, that plasma then is called interstitial fluid. So basically interstitial fluid is just, again, plasma that has left the capillaries and is now surrounding the body's tissues. From there, that plasma can actually go from the tissue and kind of seep into the lymphatic system. And so remember we said that the fluid that you find flowing through the lymphatic system, it's called lymph, and that's basically how that lymph gets into the lymphatic system. It's because of plasma leaking out of the capillaries and bathing the tissue and then making their way into the lymphatic vessels. Not only can the plasma leak out of the capillaries and enter the lymphatic system, the white blood cells that are in the capillaries can also leave and enter the lymphatic system. And so there's a couple of different ways that they can do that. So there are things that could cause the cells that line the capillaries to actually increase their distance from one another, which would then allow those white blood cells to leave. And again, it would be a good idea to have white blood cells leave the capillaries, for example, if you have something infecting the surrounding tissue. And some of these white blood cells may then continue on to kind of seep into the lymphatic system as well. Another place where white blood cells can enter the lymphatic system is through the lymph nodes. So it turns out that there are vessels of the circulatory system that feed into certain lymph nodes. Those vessels are called high endothelial venules. And it turns out that here's another place where white blood cells can leave the circulatory system and then enter the lymphatic system. And in fact, they enter directly into these lymph nodes here. All right, so again, the importance of these high endothelial venules, or HEVs, is that here's where white blood cells exit in order to populate the lymph node. You're seeing here that a lot of stuff is leaving the circulatory system, right? So we've shown you that you have plasma leaving the capillaries, you have white blood cells leaving the capillaries. If this were to continue, then eventually your capillaries, your circulatory system, will end up being drained. So there's actually a way for all of this stuff to return back to the back to the circulatory system. And basically, here's how that happens. Basically, all of the lymphatic vessels that comprise the lymphatic system, they all get together and drain into this big lymphatic vessel called the thoracic duct. And from there, all of that lymph then empties into a vessel of the circulatory system that's called the subclavian vein. So that's how all of the stuff that left the circulatory system ends up getting returned to the circulatory system. And so to sum up all of this stuff that we just mentioned, so how does the circulatory system connect with the lymphatic system? The plasma that leaks out of the capillaries, they enter the lymphatic system to become lymph. White blood cells and pathogens enter the lymphatic system at several points. So we mentioned, for example, here just having them seep out of capillaries, and then most of them enter the lymphatic system by seeping out of these things called high endothelial venules directly into the lymph node. That lymph that ends up populating the lymphatic system, it ends up being returned to the blood, right? So that the plasma portion of the blood doesn't end up drying out. So just to give you a point of reference, the exact part in the body where you find the thoracic duct emptying into this subclavian vein is right here. And so this picture shows you kind of how the lymphatic system connects to the circulatory system. This part that we just showed you here, the thoracic duct emptying into the subclavian vein, is found right here in the yellow. All right, so just to give you a little point of reference. So that's again how the circulatory system connects to the lymphatic system. And so now that we've taken a look at how all of these leukocytes, all of these white blood cells, can find their way throughout the entire body, we can now start taking a look at what some of these white blood cells can do. And specifically, we'll take a look at the white blood cells that are part of the adaptive immune system. Two of the main types of cells that are involved with the adaptive immune response, again, are T cells, or T lymphocytes, and B cells, or B lymphocytes. And what causes these cells to get activated is if they see and if they bind to a foreign molecule. And it turns out that T cells and B cells bind to foreign molecules in a little bit different way. One thing that they have in common is that they both have a receptor on their surface that allows them to bind to these foreign molecules. And so if you're considering a T cell, and the molecule on the surface of the T cell that it uses to grab onto a foreign molecule, that molecule is represented by this shape right here, and it's called a T cell receptor. So again, T cell receptors are what T cells use to bind to foreign molecules. Closely associated with a T cell receptor in this picture here is represented by this purple thing. And so this purple thing is called a CD4 molecule or a CD8 molecule. And so we'll see that there are different types of T cells. Some types have a thing called a CD4 molecule. Other types have a thing that's called a CD8 molecule. But regardless if it's a CD4 or CD8, they both basically do the same thing. And what they do is that they strengthen the binding between the T cell receptor, and we'll use TCR as our abbreviation for T cell receptor. So it strengthens the binding between the T cell receptor and the foreign molecule. Right, so whatever foreign molecule this T cell is going to bind to, it'll bind right here to the T cell receptor. The role of this CD4 or CD8 molecule is to make sure that that the T cell stays attached to that antigen, to that foreign molecule, long enough so that the T cell can become activated. B cells also have a receptor on their surface that lets them bind to foreign molecules, and the name of the molecule that does this for B cell, it's not a T cell receptor because it's not a T cell. Instead, it's called a B cell receptor. And so it turns out that B cell receptors, it's a type of molecule that's generally called an immunoglobulin or an antibody. And so this T cell receptor is not considered to be an antibody, but this B cell receptor does fall under that category of antibody. And again, what this B cell receptor does is that it's what the B cell uses in order to bind to foreign molecules. It turns out that there are some other important molecules that you find on the surface of T cells and B cells, and we'll point those out here. This peach molecule that you find on B cells, it's called a CD40 molecule, and it plays a role in helping to activate this B cell. And so we'll see exactly what role that is here in a minute. Another molecule that you find on the surface, not only of B cells, but also on T cells, is this black molecule here. And what that is, it's something called a cytokine receptor. So it allows this B cell to bind to things called cytokines, kind of like this. So this yellow circle represents a cytokine, and basically what a cytokine is, these are the molecules that leukocytes, that white blood cells, use to communicate with one another. Right, so, for example, you might have macrophages that release cytokines that tell certain T cells what to do. You might have T cells that release certain cytokines that tell B cells what to do. So, in order for these different cells of the immune system to be able to get the message from those cytokines, they have to have these receptors for them. So again, a cytokine receptor, these are things that you find on all leukocytes, on all white blood cells. Another important molecule that you find on the surface, not only of B cells but also on T cells, are these things called MHC1 molecules. And so we'll see what these molecules do a little bit later on. But again, what these MHC1 molecules are, where you find them, actually not only do you find them on B cells, not only do you find them on T cells, but you actually find them on every cell in the body, at least every cell in the body that has a nucleus. And it turns out that that means every cell in the body except red blood cells. So red blood cells don't have nuclei. So again, we're taking a look at how cells of the adaptive immune response, what they do. And T and B cells are two major cells that are considered to be part of the adaptive immune response. What we're taking a look at here are some of the major molecules that you find on the surface of T cells and B cells. If you're going to make some general kind of descriptions about T cells and B cells, particularly how T cells and B cells differ from cells of the innate immune system, here are those general descriptions or general statements. So one of the things about T cells and B cells is that they actually take a little bit longer, actually in some cases quite a bit longer, to fully activate as compared to cells of the innate immune response. So if you remember a little while back, we mentioned that one of the things that makes the cells of the adaptive immune response different from innate immune response cells is that they're able to become fine-tuned. So after a T cell or a B cell first binds to a foreign molecule, it can actually undergo this kind of learning process where it over time becomes better adaptive, more able to get rid of the pathogen that it's just bound to. And it turns out that it takes time for that fine-tuning to occur. And so that's why we're saying here that it actually takes a little bit longer to fully activate T cells and B cells as compared to cells that you find in the innate immune response, because those cells of the innate immune response don't undergo that fine-tuning. Another kind of general description you can make about T cells and B cells is that they give rise to these things called memory cells. And again, this is something that the cells of the innate immune system don't do. And basically what memory cells are, they're cells that can live for a relatively long time. Right? So after an infection is over, these things called memory cells stick around just in case you happen to see that same pathogen sometime in the future. And when these memory cells do see that, they're able to activate quicker and stronger against that pathogen as compared to the T cells and B cells that are just seeing them for the first time. Right, so T cells, B cells can give rise to memory cells. Again, memory cells are cells that are descended from B or T cells that have already encountered a pathogen. And what memory cells do is that they persist after an infection is over. And what can happen to them is that they can be reactivated relatively quickly when they see that same pathogen in the future. So this is one of the major reasons why you don't get, there's some illnesses where you don't get sick from them typically twice. So for example, when you get chicken pox once, you produce these memory cells as a result of that encounter. And so if you were to see the chicken pox virus again in the future, that's one of the main reasons why you don't typically get chicken pox again, because you have these memory cells that quickly respond to that virus if it's seen again. Another general characteristic of T and B cells is that, well, we mentioned this before, they actually work closely with components of the innate immune system, so components like macrophages, dendritic cells, natural killer cells. So there's a lot of crosstalk between, again, these cells of the innate immune response and these cells of the adaptive immune response. And one last characteristic of T cells and B cells is that each type of those cells, so each type of T and B cell, recognizes a very specific part of a foreign molecule. And so here's how that's different than how innate cells respond to foreign molecules. And so if you remember cells of the innate immune system, so cells like macrophages, for example, the reason why a macrophage knows what to phagocytize, the reason why it knows what to eat, is because it's somehow able to recognize things that are probably foreign to our body. And the reason why they can recognize those foreign molecules is because they have molecules on their surface called toll-like receptors. And if you remember what these toll-like receptors do, is they can recognize what's called general molecular patterns that you tend to find on molecules that don't belong in the body, so molecules that aren't typically part of our cells. So an example of a thing that, an example of a molecule that a toll-like receptor would generally recognize as being foreign is the flagella of bacteria, right? So our cells don't have flagella on it, so if we were to encounter something that does have flagella on it, it's probably a good idea to mount some kind of response against it, because it's probably something bad like a bacterium. These toll-like receptors can recognize general patterns that you only find on flagella, for example, but not on any molecule that you find on our cells. So the way that B cells and T cells, though, recognize foreign things, it's a little bit different. So like we mentioned, they're more specific. So they kind of go beyond being able to just say, oh, I can recognize this generally as a foreign molecule. Instead, B and T cells actually recognize specific parts of this flagellum that make it foreign. Right, so here's maybe a little bit more clarification on that. So if you consider the flagella of a bacterial cell, it's a structure made out of protein, and the protein that you find in flagella, it's a type of protein called flagellin. So we'll show you a ribbon structure of flagellin. Again, the toll-like receptors on a macrophage would basically recognize this whole thing just in general and say, oh, this is a molecule that you don't typically find on our cells. It's a foreign molecule. this is good reason for me to eat this thing, so that I can potentially prevent it from causing any harm. At least the bacteria that's attached to it, I can prevent it from doing any harm. B and T cells are a little bit different in that they're more specific in which part of this foreign molecule it recognizes. So here's what we're talking about. So again, whereas this macrophage would just recognize this whole thing generally as a foreign molecule, the cells of the adaptive immune response, for example, a B cell, would do this. So it turns out that we have millions of different types of B cells. What makes them all different is that they all have slightly different B cell receptors that allow them to attach to a slightly different looking foreign molecule. So, for example, this type of B cell might have a B cell receptor that would allow it to recognize and bind to just one small specific part of this entire flagellin molecule that you find on bacteria. We have other types of B cells that are also useful in that they can recognize foreign things, but what makes it different is that it recognizes a slightly different looking foreign thing. So this B cell, for example, might recognize a different part of the flagellin protein than this B cell would. And so we have many different types of B cells. There might be another type of B cell yet that might recognize this part of the flagellin molecule. So that's what we're saying when we say that B cells and T cells, parts of the adaptive immune response, they recognize more specific portions of a foreign molecule. So to throw some terms at you, terms that are useful in understanding what occurs during the immune response. There's something called an antigen. And so for our purposes, we'll consider an antigen to be a foreign molecule that stimulates an immune response. So a molecule that's recognized as foreign by our white blood cells. And so an example of a molecule that fits this description of what an antigen is flagellin. So bacterial flagellin protein, that's something, again, that our cells don't have, so it's something that would be recognized as foreign. So it's an example of an antigen. What we're saying here, though, is that an antigen can be a relatively big molecule, and so there are specific portions of an antigen that can be recognized by different types of B cells and T cells, because B cells and T cells are more specific in what they recognize. So when you're talking about the specific and smaller parts of an antigen that can be recognized by different types of B cells and T cells, each one of those smaller parts is called an epitope. Right? So in words, epitope is, again, the specific smaller part of an antigen that's recognized by B or T lymphocyte. And so, for example, this whole thing, again, is considered an antigen. Here's one specific epitope of that antigen that happens to be recognized by this type of B cell. Here's another specific epitope on the same antigen that's recognized by a different type of B cell. Here's yet another kind of epitope that you find on this antigen that happens to be recognized by this type of B cell. So one single epitope can have, one single antigen can have many different epitopes on it that can be recognized by different types of B cells and T cells. And so we'll throw that down here in words so that you can jot that down. Basically, the reason why the different types of B cells and T cells can only recognize certain types of epitopes is because if you take a look at the shape of the B cell receptors, and if you take a look at the shapes of the T cell receptors, they're all slightly different. So here's what we mean. If you take a close-up look at these B cell receptors, for example, you'll see that the ends of them have slightly different shapes. So I drew it kind of cartoony schematically here, but the idea is the same. The ends of those B cell receptors have different shapes, which means that each type of B cell receptor can only bind to a certain shaped epitope. So this guy wouldn't be able to bind to the same thing that this guy would because of the different shapes. And so that again explains why each type of B cell and also each type of T cell only recognizes one specific type of epitope. It's because of the unique shapes of the receptors that you find on B cells and T cells. All right, so the point here is that B cells and T cells recognize more specific parts of a foreign molecule as compared to cells of the innate immune response. It turns out though that if you compare how a B cell recognizes an epitope to the way that a T cell recognizes an epitope, there's actually some differences there, and so we'll take a look at that next. All right, so again, B cells and T cells recognize epitopes in different contexts. Here's what that means. If you consider the way that a B cell recognizes an epitope, here's basically how it does so. If there's, for some reason, a foreign molecule or an epitope that's free-floating in the body, so for example in the blood or maybe in the lymphatic system, it turns out that B cells are able to directly bind to that epitope and then likely get activated as a result of it. It turns out that T cells recognize epitopes in a little bit different way. So they can't bind to free-floating epitopes like a B cell can. Instead, a T cell will bind to an epitope only if another cell in our body has come across a foreign thing and is presenting then that foreign thing to this T cell. So in pictures, a T cell will only recognize an epitope, again, if some other cell in the body, for example, maybe a macrophage, has come across a bacterial cell, for example, taken that bacterial cell in and is now showing off a piece of that bacterial cell to the outside. So then and only then only if a cell is presenting an epitope to the outside would a T cell be able to recognize it and likely get activated as a result. And so here's how I kind of look at this in order to remember it. So it turns out that the T in T cell and the B in B cell stand for something. T stands for thymus because that's where T cells mature. B stands for bursa fabricius, which is the part in birds where B cells actually mature. So that's what T and B officially stand for. But the way I see it, B stands for buffet because that kind of describes how B cells go about binding to their epitope. So if you think about what happens when you go out to eat at a buffet, you don't have to wait to get served, right? So you have a table assigned to you, then you get up and you grab your own food. B cells basically do that. They can grab their own epitope. T in this instance stands for tray, because T cells will only bind to an epitope if that epitope is being presented to it on a tray by another cell. So that's kind of one quick way of remembering it. So to write it down, to make it more official, B cells, what makes them, one of the things that makes them different from T cells is that they bind directly to free-floating epitopes. On the other hand, T cells will bind to epitopes only if it's presented to it by another cell. And so what we'll do next is take a closer look at this interaction between a T cell and the cell that's presenting the epitope to it. And so in particular, we'll take a look at all of these molecules here that are involved in that presentation. So we'll we'll make bigger this particular interaction so that it looks like this. So again, you're taking a look at the bottom here, the T cell. Here is the molecule on the T cell that lets it bind to the epitope. And if you remember, that molecule is called a T cell receptor. This red thing represents the epitope that the T cell is binding to. Here up here is the cell that is presenting that epitope to the T cell. And you'll notice that this cell is using this brown molecule here in order to present that epitope to the T cell. So the name of this molecule that presents epitopes to T cells, it's called an MHC molecule. And again, this purple thing here, if you remember, that's called a CD4 or CD8. So depending on the type of T cell it is, this purple thing will either be called a CD4 molecule or a CD8 molecule. And again, if you remember, we said that the function of this CD4 or CD8 molecule is that it helps to strengthen the interaction between the T cell receptor and the epitope. And so you can see more closely how that's done here. So basically this T cell receptor is kind of an extra arm that the T cell uses to kind of grab a hold on to that MHC molecule, and it ensures that that MHC molecule / epitope stays together with the T cell receptor long enough in order for the T cell to become activated. And so we're saying here that T cells again are different from B cells because they only recognize epitope if it's being presented to it by an MHC molecule. And basically the reason why that is because the molecule that T cells use to grab onto epitopes, this T cell receptor, it actually recognizes not just the epitope, but it's kind of built to recognize an epitope in combination with an MHC molecule. So you kind of see that here. This T cell receptor is kind of binding to both this epitope and this MHC molecule at the same time. In other words, this T cell receptor is kind of built, again, to be activated only if it's binding to MHC and epitope at the same time. So we'll jot that down here to make it official. T cells bind to epitopes only if it's presented by another cell. The reason why that is because T cell receptors recognize the epitope and MHC molecule as a combination. Alright, so we've just said that T cells and B cells, we have millions of different types of B cells. We have millions of different types of T cells. Each one of them recognizes one specific type of epitope. And that's what ends up activating T cells and B cells. Before we take a look at what happens after T cells and B cells get activated, we'll take kind of a step back and say a little bit something more about antigens. And so remember, antigens are just the way that we're going to define the term antigen is it's a molecule that stimulates an immune response. It's something that our white blood cells can recognize as a foreign thing. And so examples of places where you would find such antigens, so we've already said that bacteria have antigens on them. Specifically, we looked at flagella as an example of an antigen. But there's all kinds of other molecules that you find on bacteria that normally serve some normal purpose for the bacterial cell. But if they make their way into our bodies, our white blood cells should recognize these things as foreign things because you don't normally find them on our cells. So for example, there's all kind of proteins that you find in the membrane that would be considered antigens. There's portions of the capsule that are considered to be antigens. Plasmid DNA actually is considered to be an antigen, again, because all of these things should be recognized as foreign. They're things that are not normally found on our cells. Not only do you find antigens on bacteria, but viruses also have antigens on them. So this happens to be a schematic of the HIV virus, and so there's all kinds of molecules that are in the envelope of the HIV virus that should be recognized as foreign. In other words, these are molecules in the envelope that are considered to be antigens. There are also molecules inside the envelope that would stimulate an immune response if our white blood cells see them. So viruses have all kinds of molecules that are considered antigens. Bacteria have all kinds of molecules that are considered antigenic. Fungi, different types of parasites, all of those things have molecules that you wouldn't normally find on our cells and therefore would be considered antigenic. So there's many types of molecules that are considered antigens out there, but you can categorize them all as being just one of two types of antigens. And so all antigens out there are considered to be either exogenous antigens or endogenous antigens. You might know that exo means outside, endo means inside. So this gives you a clue as far as where you would find, or at least where these antigens, these different types of antigens, are made. And so to show you the difference between what an exogenous antigen is and what an endogenous antigen is, what you're taking a look at here on this screen is a bacterial cell, and these are some cells that are found in our own body. We've just seen that bacteria produce all kinds of molecules that serve some purpose normally for that bacterial cell, but if our white blood cells end up seeing them, our white blood cells should recognize those as foreign molecules and mount an immune response against it. So whenever you're talking about antigens like these, ones that are made by cells outside of our body, in other words, ones that are made by foreign cells, those are what's considered to be exogenous antigens. And so again, here is its definition in words: molecules made by foreign cells that stimulate our immune system. It turns out that there are instances when the cells in our own body might actually make molecules that look weird, that look foreign. Right? So some of those molecules that the cell makes might stay inside of it. Some of them might be on the surface. But if it's a weird-looking molecule that's produced by one of our own cells, that's what would make it an endogenous antigen. Again, endo means inside because endogenous antigens, again, are molecules made by our own cells that would stimulate an immune response. And so you might be wondering, well, why would our own cells ever make weird-looking molecules that might be recognized as foreign. Well, if you remember from a couple of weeks ago, one of the things that might cause that is if our cells are infected with a virus, right? Because if a virus ends up getting inside of our cells, one of the things that viruses can do is basically hijack the machinery of our cells, right? They can take over the ribosomes, for example, and they make those ribosomes produce viral proteins instead of normal proteins that you normally find inside of our cells. So that's an example of when our own cells might start making weird-looking proteins, is if it's infected with a virus. Another example of a time when our own cells might produce weird-looking proteins is if it's been transformed into a cancer cell. Right? So cancer cells, turns out that they make proteins that our normal cells would not make. So that's, again, another instance when our cells would end up producing endogenous antigen. So what we'll do is this. So next we'll take a look at how our immune response, or how we respond to exogenous antigens, and then a little bit later on we'll take a look at the immune response to endogenous antigen. So again, first, how do we respond to exogenous antigen? One type of cell that plays a really big role in responding to exogenous antigen is a type of cell called a dendritic cell. And it turns out that technically dendritic cells are categorized as being part of the innate immune response. But it turns out that dendritic cells work really closely with parts of the adaptive immune response. Specifically, we'll see that they work really closely with a type of T cell called a helper T cell in order to respond to exogenous antigen. Right, so remember, before we said that there's a lot of crosstalk, there's a lot of cooperation between parts of the innate immune response and parts of the adaptive immune response. And so here's an example of that. So again, we'll see that parts of both innate immunity and adaptive immunity work together to respond to exogenous antigen. So before we see what dendritic cells do, we'll mention what you find on dendritic cells. So it turns out that dendritic cells, just like macrophages, have toll-like receptors on their surface that allows them to recognize general patterns that you find on foreign molecules. Not only that, but dendritic cells also have actually two different types of MHC1 molecules. So dendritic cells have MHC1 molecules, and dendritic cells also have a type of molecule called an MHC2 molecule. So it shouldn't be surprising that dendritic cells have MHC1 molecules, because we've already mentioned before that you actually find MHC1 molecules on every cell in the body except for red blood cells. It turns out that MHC2 molecules, you don't find those on every cell in the body, and it turns out that there's only certain types of white blood cells that have these MHC2 molecules on them. And so an example of a type of white blood cell that has that are these dendritic cells. So that's what dendritic cells have. We say that these cells play a big role in responding to exogenous antigen, basically because these are one of the first types of white blood cells to contact exogenous antigen. So if you think of all the different types of white blood cells out there, chances are it's going to be a dendritic cell that makes first contact with exogenous antigen. And basically the reason why is because, if you remember, exogenous antigens are antigens that you tend to find on things like bacteria. There are certain parts of the body where bacteria tend to gain access. So places like the respiratory tract, right, you can breathe bacteria in. There's the gastrointestinal tract, right, you can eat bacteria. So those are common places where bacteria get access to the body. And it turns out that dendritic cells, they tend to hang out in those areas of the body. Right, so dendritic cells are found in the areas that are the usual entry points for pathogens. So places like the skin, the nose, the lungs, the intestines. And basically the job of the dendritic cell is that basically they run surveillance for the adaptive immune system. So again, dendritic cells hang out in these parts of the body. When they're in these parts of the body, dendritic cells run across all kinds of molecules, right? So some of those molecules might actually just be normal molecules that our healthy cells produce. Other ones of those molecules might actually be bad molecules, so ones that are produced by bacteria that are going to try to infect us. So basically what dendritic cells do is that all of those molecules that it runs across, they're continuously taking it in and then showing it off to a type of T cell called the helper T cell. And basically what its job is to let that helper T cell decide whether this molecule that it's run across is worth responding to, right? So it's going to trust the T cell to be able to tell the difference between a bad molecule, one that you normally find on bacteria, for example, and a good molecule, so one that would normally be made by our healthy cells. So in words, what dendritic cells do is that they kind of see them as running surveillance for the entire immune system. So they continuously monitor an area for suspicious activity. And what they do more specifically is that they help T cells decide whether to become activated or not. Right? So if there's something bad that enters the body, the dendritic cell will help the T cells decide whether they should become activated as a result of that molecule. So here's how that looks. So again, when the dendritic cells are hanging out in the nose, the skins, the lung, the intestines, it's always running across these molecules that you find in those places. Some of them might be good, so some of them might be molecules that our healthy cells would normally make. Some of them would be bad. And basically the job of the dendritic cell is to show these things off to a helper T cell and let it decide whether it's something that's worth mounting an immune response to. So if a dendritic cell happens to run across an OK molecule, it's kind of not smart enough to figure out on its own whether that's good or bad. It'll just take in any molecule that it runs across. And what it does after it takes in that molecule is that it chops it up for the purpose of loading one of those pieces onto this MHC2 molecule. After that's done, it exports that entire thing to the outside. And so basically what the dendritic cell is doing now is it's saying, look, I've run across a molecule. I'm not really smart enough to decide whether it's a good or bad molecule. Mr. Helper T cell, why don't you decide whether or not this is something that's worth mounting an immune response to? And so that's what happens. So whenever a cell, like a dendritic cell, is showing a molecule off on the outside via an MHC2 molecule, what occurs is that a helper T cell, that's the specific type of T cell that recognizes things that are loaded on to an MHC2 molecule. And so the helper T cells that you find in the blood and the helper T cells that you find circulating in the lymphatic system, they should all have T cell receptors that will only bind to and only respond to foreign things. So if a helper T cell happens to run across a good molecule, so a molecule that's normally found on our cells, that's being presented to it by an MHC2 molecule, it should be kind of programmed. It should be smart enough because of the shape of its T cell receptor to not bind to and therefore not respond to that molecule and kind of leave it alone. It ends up not activating other parts of the immune system. Right. So in words, whenever this occurs, that helper T cell does not get activated if the dendritic cell is showing it an OK, in other words, a non-harmful molecule, one that you would normally find on our cells. And so you see on this drawing of a helper T cell, there's this blue molecule on here that we haven't shown you before. And so we'll just mention now that the name of that blue molecule, it's called a receptor for a co-stimulatory molecule. And so we'll see what role this molecule plays here in a minute. But the point we're trying to get across here is that if a dendritic cell is showing off something that is actually not harmful to a helper T cell, that helper T cell should know better not to mount an immune response against it. If on the other hand this happens, so say that there is now a different dendritic cell that's hanging out in the nose, the skin, the lungs, the intestine, and it happens to run across not a good molecule, but one that's bad, so one that you might find on pathogenic bacteria, for example. Here's how the response differs. So again, what this dendritic cell will do is that it'll take in that bad molecule. It basically takes in everything that it runs across. But it's different in that if it's a bad molecule, so if it's one that you typically find on bacteria, remember that dendritic cells have these toll-like receptors that actually can grab a hold onto molecules that you typically find on bacteria. And so whenever that occurs, if this is a bad molecule that the dendritic cell is running across and it binds to that toll-like receptor, it causes this dendritic cell to do something that it didn't do here. Right? So whenever something binds to the toll-like receptor of the dendritic cell, it causes that dendritic cell to make a molecule called a co-stimulatory molecule that it ends up exporting to the outside. Right? So again, this blue thing is called a co-stimulatory molecule. So that's what happens whenever a dendritic cell has its toll-like receptor engaged, and again, it's engaged only if it runs across a molecule that you tend to find on foreign cells. After it runs across that molecule, again, that dendritic cell takes in that molecule, chops it up to bits, and it loads one of those bits onto an MHC2 molecule, and again, it exports that whole thing to the outside. And so again, you see the difference here. If a dendritic cell runs across a bad molecule, not only will it show it off to the outside via an MHC2, but it also expresses this thing called a co-stimulatory molecule. So now if a helper T cell happens to run across a dendritic cell that's doing this, there's a couple of reasons now for this particular helper T cell to get activated. One, it's actually seeing a molecule that it should recognize as actually a foreign thing. And another thing that it's seeing is this dendritic cell is expressing a co-stimulatory molecule, and helper T cells have a receptor that can bind to that molecule. And again, a dendritic cell would only express a co-stimulatory molecule if it's had its toll-like receptor engaged, and its toll-like receptor can only engage things that you typically find on foreign cells, right? So this helper T cell again has a lot of reason now to get activated, because it's seeing a lot of proof that there actually is something harmful that's floating around. Right, so again, in this case, if the helper T cell sees these things, that's what causes the helper T cell to become activated. And again, it happens only if this presenting cell, in this case a dendritic cell, shows it both a foreign antigen and a co-stimulatory molecule. And again, this co-stimulatory molecule is expressed by that dendritic cell only if its toll-like receptor is engaged. Some other things that we can say to kind of sum up the main points of this slide is this. So we mentioned that there's this molecule called MHC2 that plays a big role in responding to exogenous antigen. And so specifically, again, what MHC molecules do is that they present molecules that have been phagocytized and then chopped up by things like dendritic cells. And what MHC2 molecules do is that they present to a specific type of T cell called a helper T cell, and we'll use TH as our abbreviation for helper T cell. And then another thing to mention here as a take-home point is that speaking of helper T cells, one of the ways that you can identify a helper T cell is that they have these things called CD4s on their surface. So you remember from a little bit back, we mentioned that all T cells have this molecule that helps it to stay bound to the MHC molecule. We said that this molecule is called CD4 or CD8, depending on the type of T cell that it's found on. So what we're saying here is that if you're talking about a specific type of T cell that's called a helper T cell, the type of CD molecule that it would have on its surface is a CD4 molecule. So helper T cells don't have CD8, they have CD4s instead. So what we just said is that if we happen to encounter exogenous antigen, these dendritic cells again would likely be the first type of cell that will run across it. It'll take it in, show it off to a helper T cell in order to activate it. So whenever we say that a helper T cell becomes activated, here's what we mean. So we'll kind of redraw this so that we're showing again a dendritic cell that's showing an exogenous antigen off to a helper T cell. When it becomes activated, what we mean is that it makes copies of itself. So it makes clones of itself. And some of those clones become these things called effector cells. So in this case, these would be called effector helper T cell. And basically, whenever you see this term effector as part of the cell's name, that means that that particular cell is ready to perform whatever its function is. So these guys are ready to do what helper T cells do, which we'll see is to help activate other parts of the immune system. So what you're taking a look at here is just the start of the immune response to exogenous antigen. So nothing has really been done so far here that would potentially kill the bacteria that possesses these exogenous antigens. This is just kind of laying down the foundation. And so another type of cell that is involved in responding to exogenous antigens are B cells, which again are a type of cell that's considered to be part of the adaptive immune response. It turns out that B cells, and we're showing it here, this is another example of a type of white blood cell that has MHC2 molecules. And so you might be able to guess that B cells, it's another type of cell that can show things off to a helper T cell as a result of having this MHC2 molecule. And so here's what B cells can do. Remember we said that B cells are different than helper T cells in that B cells are able to directly bind to free-floating antigen. So if it happens to run across an antigen that is bad, and it might do this in a lymph node, for example, that B cell now is just itching to respond, right? Because this B cell now has, again, encountered a foreign molecule. That foreign molecule likely means that there's a bacteria that made it, and that bacteria might be harmful to us. It turns out, though, that B cells, they can unleash all kinds of destruction whenever they get activated. And so before this B cell responds to this foreign molecule that it's bound to, it's going to make doubly sure that what it's responding to really is a bad thing. It doesn't want to unleash all of the destruction that it can unleash on something that's actually not a bad thing. And so because of that, B cells, before they act, in a lot of instances, get confirmation, right? So they basically ask another cell of the immune system to make doubly sure that it's bound to something that's bad before it actually responds to it. And so here's how it does it. So that, again, that B cell, before it actually gets activated, it takes in that antigen that it's just bound to, and it chops it up, and it loads one of those pieces onto one of its MHC2 molecules, and then that whole thing gets exported to the outside. And so basically what this B cell is doing is that it's saying, look, I've run across something, I'm pretty sure it's a bad thing, I'm pretty sure it's a foreign molecule, but just to make sure, before I respond to it and unleash all kinds of destruction, I'd like someone out there to just confirm to me that this really is a bad thing. And so it turns out that the cell that does the confirming is one of these helper T cells that have become activated by the dendritic cell that presented antigen to it. Right, so this antigen that activated this helper T cell should be the same as this antigen that this helper T cell is going to respond to. Right, so if there's a helper T cell around that's activated, and it sees this antigen that's being presented by that B cell, that helper T cell will go, oh yeah, I also recognize this as a foreign molecule. This probably means that there's something bad around. B cell, you best get activated to help get rid of that potentially harmful bacterium. And so once that helper T cell recognizes and confirms to the B cell that this really is a foreign antigen, again, it ends up activating the B cell. Here's how it activates the B cell. It actually ends up producing a molecule that can bind to the CD40 molecule that you find on the B cell, like this. Not only that, but once this helper T cell recognizes this as a foreign molecule, it also produces cytokines that are able to bind to the cytokine receptor that you find on the B cell. So these are two things that the helper T cell does in order to help activate this B cell that's run across something that's foreign. Right, so this B cell again becomes activated as a result. So to sum up what we're saying here, how about if we do this? We're saying that B cells, these are another key cell that responds to exogenous antigen. They can recognize free-floating exogenous antigen. Before responding, though, to that antigen that they attach to, they first get confirmation from one of these activated helper T cells. And here's how they get that confirmation. So that B cell takes in the antigen that it's bound to, and it chops it up. And so we'll use this AG prime, this AG apostrophe as our shorthand for antigen. So it takes an antigen, chops it up. The B cell then takes one of those fragments and presents it via an MHC2 molecule to a helper T cell. And so if the helper T cell then sees that antigen and agrees that it is indeed a foreign molecule, it ends up activating that B cell. And again, it ends up activating that B cell by doing two things. So one thing is that the helper T cell produces a CD4 ligand, and our abbreviation for CD4 ligand is going to be CD40L, L for ligand. And so to relate that to this picture again, this peach molecule that you find on the B cell, that's called CD40. This molecule that the helper T cell produces, if it agrees that this is in fact a foreign molecule, that's called the ligand for CD40, or it's CD40L. In addition to producing that CD40 ligand, if that helper T cell recognizes this as a foreign molecule, it also produces cytokines to activate the B cell. So we'll jot that down here. Right. So again, keep in mind that all of these events that you're seeing here, they're all connected. So whenever we see an exogenous antigen, a dendritic cell will likely see it first. It'll activate a helper T cell as a result. One of those things that those activated helper T cells can do is confirm to a B cell that the thing that it has bound to is foreign. And so the helper T cell then ends up activating it. To give you an idea of where all of this is taking place, remember we said that these dendritic cells are found in places like the skin, the nose, the lungs, and the intestines. So that's where these dendritic cells run across these molecules. After they run across these molecules, they end up traveling to the nearest secondary lymphoid organ, and that's where they end up presenting that molecule to a helper T cell, and that's where these activated helper T cells end up being produced. These B cells, basically that's where they meet up with the helper T cell. So these B cells run across antigen as they're traveling around in the lymphatic system, or perhaps even the blood. In order to get that confirmation, they have to meet up with an activated helper T cell in one of the secondary lymphoid organs like the lymph node. And so that's where this interaction occurs. And again, if the helper T cell confirms that it indeed is a foreign antigen that the B cell is showing it, then it releases the cytokine and it shows off the CD4 ligand. And again, all of this is happening in the lymph node, or a different secondary lymphoid organ. So we're saying here that after this confirmation has taken place, the helper T cell helps the B cell to become activated. And so what we'll show you next is what happens when a B cell becomes activated. And so it's actually the activation of the B cell then that causes things to directly happen in order to get rid of the exogenous antigen, or at least the bacterium that produced the exogenous antigen. So here we go again. Here's a B cell that's encountered an antigen. It's gotten confirmation from a helper T cell that it indeed is foreign, so it activates that B cell. When we say that a B cell becomes activated, it means that it makes copies of itself. So it ends up making clones of itself. Some of those cloned B cells differentiate into a type of cell called a plasma cell. Some of the other cloned B cells differentiate into a type of B cell called a memory B cell. And we've already mentioned a little something about memory cells, and so we'll mention it again. The reason why these memory cells are so valuable is because they stay behind. So they remain in the blood and also in the lymph after that infection is over. So whatever bacterium is producing this exogenous antigen that's causing all of this response, these memory B cells will stick around after that bacterium has been taken care of. And again, if these memory cells happen to see that same antigen in the future, they are able to quickly differentiate into strong plasma cells that can then quickly get rid of that antigen. And so we'll say a little bit more about this later, but when these memory B cells see that same antigen in the future, the plasma cells that they become are better equipped to deal with that antigen as compared to these kind of first-line plasma cells that are produced when our body sees that antigen for the first time. So again, these memory cells are really valuable. They, for the most part, prevent us from getting sickened with the same pathogen in the future. These plasma cells are valuable for a different reason. And so what plasma cells are, they're basically antibody-producing factories. Right? So once plasma cells are available, what they do is that they just produce and secrete a whole bunch of, well, let's say this first. So one of the reasons why, going back to the memory B cell, one of the reasons why these memory cells are a lot more effective against an antigen as compared to the B cells that see the antigen for the first time is because if you compare the number of cytokine receptors on the surface of memory B cells as compared to this B cell that got the whole thing started, there are a lot more of these cytokine receptors on the surface of memory B cells. So it makes it a lot easier then to activate that memory B cell as a result of that. So then again, going to these plasma cells then, so the reason why these plasma cells are so valuable is because they get rid of the infection that's happening right now. And the way they do that is that they produce and secrete a whole bunch of antibodies. Another term for antibody is immunoglobulin. So you might see those two terms being used interchangeably. And basically what antibodies or immunoglobulins are is that they're proteins produced by, again, plasma cells that just stick to stuff, right? So this plasma cell was derived from this B cell. So whatever antigen that this B cell attached to will be the same antigen that these antibodies should be able to attach to. Right, so if there's one bacterial cell around that produced exogenous antigen that caused all of this activation to occur, there's probably other bacteria of the same type around, and that's who these antibodies would go after. Right, so say that here's the bacterium that actually started the whole thing, the bacterium that caused the B cell to get activated. These antibodies that are being produced by this plasma cell, which came from that B cell, then would be able to stick to the antigen that's found on that bacterial cell, so the exogenous antigen that started this whole process. And once antibodies have stuck to a bacterial cell, now's when stuff actually happens that causes the elimination of that bacterial cell. So we'll point out those things next. So what happens after antibodies stick to a bacterium? Why would that cause a bacterium to be eliminated? So one of the things that occurs as a result of our antibodies sticking to a bacterial cell is that it could prevent that bacterial cell from binding to our cells and invading our cells. So for example, if this molecule that this antibody is binding to is normally involved in helping this bacterium bind to our host cell, now that there is an antibody stuck to it, it'll prevent that molecule from interacting with our host cells, which means that the bacterium would never be able to get inside of it or infect it. So how about if we just make a list here of what all antibodies can do after they bind to a bacterial cell. So antibody, so AB kind of prime, that's going to be our shorthand for antibody. Antibody binding may result in this thing called neutralization, which we just showed you is when antibody binds to a pathogen and it prevents that pathogen from binding to our cells. Another thing that can happen after antibodies stick to a bacterial cell is this, and it's actually something that we've mentioned last week. So it turns out that this is one of the things that can kickstart the complement cascade. So complement proteins can actually bind to this Fc portion of the antibodies, and that could then cause the complement cascade to start. And we mentioned last week that the complement cascade results in different things that can end up eliminating that bacterial cell. So that's another thing that can happen after antibodies stick to a bacterial cell, is that it can cause complement proteins to become activated. Yet another thing that can happen after antibodies stick to a bacterial cell is that it can cause other cells of the immune system to become activated. And so an example of a cell that can become activated if they happen to see bacteria that are coated with antibodies is this thing called a natural killer cell. And so it turns out one of the reasons why that is because if you take a look at the surface of natural killer cells, they have a receptor on them that allows it to bind to antibody that has stuck to a bacterium. All right, so the name of this receptor, it's called an Fc receptor because this portion here of the antibody is called the Fc portion of that antibody. And again, things about the Fc receptor, you find this Fc receptor on certain white blood cells, including natural killer cells. And what that Fc receptor does is that it allows the white blood cell to bind and respond to antibodies that have bound to an antigen. So here's what that looks like. If a natural killer cell runs across a bacterium that has antibodies coating its surface, because it has an Fc receptor, it's able to bind to that antibody, and that ends up activating that natural killer cell. And an activated natural killer cell can actually release substances that cause lysis of that bacterium. And so we can add this to our list then. So yet another thing that can happen after antibodies stick to a bacterial cell is this thing called ADCC, which we just described here. ADCC stands for antibody-dependent cell-mediated cytotoxicity. And so that's kind of a mouthful, but if you break it down, it actually describes really well what's happening here. Right, so ADCC is basically when antibodies stick to a bacterium, and that causes other cells of the immune system to attack that bacterium. If you break down this term, ADCC, here's what that stands for. We'll go backwards. So this last C in ADCC stands for cytotoxicity. Literally, cytotoxicity means that a cell is dying. And so that's why that's appropriate for what's occurring here, because you have a cell, in this case a bacterial cell, that's dying. The second to the last C stands for cell-mediated. So cell-mediated cytotoxicity basically means that one cell is causing the death of another cell. And so that's what's happening here. One cell, this natural killer cell, is causing the death of another cell, this bacterial cell. AD stands for antibody-dependent. So this means that this cell will only kill this other cell if antibodies have already stuck to it. And so again, that's what we see is happening here. So ADCC describes this whole process that we've just taken a look at. And so we'll mention a couple of more things that can happen after antibodies stick to a bacterial cell. So you might remember that each one of these antibody molecules has two portions that can actually bind to antigen, right? So if you consider an antibody molecule to look like a capital Y, each one of these short arms of the Y is capable of binding to an antigen. And so because each one of these antibody molecules can bind potentially to antigens, you could see something like this happening, where you get a whole bunch of bacteria kind of getting stuck together like this. And so this is a good thing for us, one, because it would prevent the spread of the bacterium, and then another reason why is because potentially a macrophage or some kind of phagocyte can come by and eat all of these bacteria all at once. So it makes for more efficient phagocytosis, basically. So this whole process here, where a whole bunch of bacteria can get glommed together as a result of antibody sticking, that's called agglutination. And then one last thing that can happen after antibodies stick to a bacterial cell is that it could actually increase the chance that this bacterial cell will get phagocytized by a macrophage. And so here's the reason why. So macrophages, even if there were no antibodies stuck to this bacterial cell, a macrophage could still potentially take in this bacterium. And the reason why is because macrophages have these toll-like receptors that recognize and bind to things on the surface of bacteria like flagella, and that tells the macrophage that it's a foreign thing, so that it ends up ingesting it. So even without, again, antibodies, the macrophage could still potentially eat this bacterial cell. But if a bacterial cell has antibodies coating it, then it just makes it even more likely that the macrophage would eat it, because it turns out that macrophages are another type of cell that have these Fc receptors. So now there's basically two reasons why this thing would be eaten by the macrophage. One is because it has foreign molecules on it that you normally find on the bacteria, represented by these red things. And also now it has antibodies binding it, and these antibodies can be bound by this Fc receptor that you find on the macrophage. So if there's antibodies coating this bacteria, again, there's double the reason now to eat that bacterial cell. In other words, the macrophage's toll-like receptor and Fc receptor can both be engaged. It turns out that whenever the macrophage's toll-like receptor is engaged, like we saw with the dendritic cell, it causes the macrophage to produce this molecule called a co-stimulatory molecule, and then it it exports it to the outside. What macrophages do after they encounter something foreign is that they end up taking it in and digesting it. And so that's a good thing, because now we've just gotten rid of a bacterial cell that might be trying to cause us harm. But it turns out that the macrophage isn't happy in just killing that bacterium. It turns out that the macrophage, after it does that, ends up activating even more parts of the immune system. So it ends up amplifying the immune response against any other bacteria that might still be out there. And the way that it does that is by doing this. So it turns out that, well, how about if we say this? So this phenomenon that we just described here, where antibody binding to a bacterial cell makes it more likely that that bacterial cell will become phagocytized, that's called opsonization. And so here again it is in words, when coding with antigen, with a protein, for example an antibody, when that increases its chances of becoming phagocytized. And again, this macrophage, after it's taken in and killed the bacterial cell, it doesn't rest on its laurels. So it actually does more to help activate other parts of the immune system. And here's how it does that. So after it chops up that bacterial cell, it ends up loading up one of those pieces onto one of these MHC2 molecules. So now we're seeing that macrophages are a third type of cell that has MHC2 molecules. So we've already seen that dendritic cells have MHC2 molecules. We've seen that B cells have MHC2 molecules. Now we're showing you that macrophages are a third type of cell that has MHC2 molecules. And so if you remember, what MHC2 molecules do is that they present antigen to a particular type of cell, a helper T cell. And so again, what this macrophage is doing is that it's saying, look, I've run across a bacterium. I know that it's a foreign thing because it's engaged one of my toll-like receptors, and so therefore I'm making a co-stimulatory molecule. Here's the actual bacterium that I've just conquered. Other parts of the immune system, you should now get activated as a result of that. Here's my proof that there's something bad going on. And so again, the specific type of cell that the macrophage can activate is a helper T cell, because helper T cells recognize antigen that's loaded onto an MHC2 molecule. After that helper T cell sees both that antigen and that co-stimulatory molecule, it becomes activated. So it ends up making clones of itself. Some of those clones become these things called effector helper T cells, which again just means that these are T cells that are ready to perform their function. And basically what helper T cells do is this. So we've already mentioned in passing before that the basic job of a helper T cell is that they help other cells of the immune system to become activated. So that's why they're called helper, because they help other cells of the immune system become activated. And so here we'll show you some examples of other white blood cells that rely on helper T cells in order to activate, or in order to become activated. Right. So examples of cells that a helper T cell can then activate after the helper T cell becomes activated itself include, we've already seen that helper T cells play a role in helping to activate B cells. So B cells for sure rely on helper T cells. Other types of cells that are activated by helper T cells include macrophages. So macrophages have cytokine receptors on them that allow them to receive the message that a helper T cell might be shouting out. It turns out that macrophages, so let's do this. So even before a helper T cell shouts out, gives a shout out to a macrophage would still be able to phagocytize a bacterial cell because it has these toll-like receptors that'll help it to recognize that the bacterial cell is a foreign thing. What happens though whenever a helper T cell activates it is that it basically gives a macrophage other abilities. So now, in addition to being able to phagocytize a bacterial cell, now that this macrophage has received the cytokine message from a helper T cell, it's now able to also release these substances, for example nitrous oxide, that is actually toxic to bacteria. So the macrophage acquires another thing, another weapon that it could use against bacteria. So in words, effector helper T cells, they activate macrophages so that they produce actually nitric oxide, not nitrous oxide, and that stuff is actually toxic to bacteria. So an activated macrophage, not only can it phagocytize something, but it can actually also release things that kills bacteria. Another type of cell that relies on helper T cells in order for them to become activated are natural killer cells. So it turns out that in order for this guy to work, it has to have become activated by cytokines that were released by a helper T cell. And then a fourth type of cell that we'll mention here is a type of cell called a cytotoxic T cell. We'll see what they do a little bit later on, but we'll just mention for now that this is a type of cell that relies on activated helper T cells in order for them themselves to become activated. And so we're seeing here that helper T cells are really, really important. They're like, I kind of see them as the generals of the immune system, because they tell all the other parts of the immune system what to do. If for some reason a person were to lose all of his helper T cells, that would be disastrous, because it would mean that none of these other cells of the immune system can ever become activated, and that's one of the reasons why HIV is such a terrible disease, because the HIV virus, the cell that it actually infects are these helper T cells. So whenever these helper T cells go away as a result of that infection, it means that all of these other cells end up not being able to become activated. So again, helper T cells, they help to turn on all of these different cells of the immune system. It turns out though that a single helper T cell typically isn't able to activate, for example, all four of these different types of cells by itself. Right? So another way of saying that is that there are different populations, there are different types of helper T cells. Some of those types of helper T cells tend to activate B cells and macrophages, for example, whereas other types of helper T cells tend to activate cytotoxic T cells and natural killer cells. And yet another way of saying that is that the cytokine that ends up activating B cells are different than the cytokines that end up activating natural killer cells. And so one type of helper T cell would release the type of cytokine that would activate these guys. Other types of helper T cells would release the types of cytokines that would end up activating these guys. Right, and so because there's different types of helper T cells, and again each type of helper T cell tends to activate a certain type of immune cell, those different types of helper T cells are given names. So we'll show you that next. So whenever you're talking about a type of helper T cell that tends to release cytokines that activate natural killer cells and cytotoxic T cells, that type of helper T cell is something called a Th1 helper T cell. So Th1 are helper T cells that again tend to activate these guys and these guys. On the other hand, you have a different type of helper T cell called Th2. So helper T cells that tend to activate B cells then aren't Th1, but they're considered to be Th2 helper T cells. So the bottom line is that helper T cells, they function in activating the other cells of the immune system, but you can categorize helper T cells based on what specific types of cells they help activate. So that's basically the story on how our immune system responds to exogenous antigens. And again, for example, you find exogenous antigens on bacteria. What we'll do next is just say some miscellaneous things about the immune response to the exogenous antigens. So miscellaneous things to note about the immune response to exogenous antigen. So one miscellaneous thing to note is that sometimes B cells actually do not need confirmation from helper T cells in order for them to become activated. So we've already seen that in a lot of cases, after a B cell binds to an antigen, one that it knows it's foreign, in order for that B cell to become activated, what we've shown you so far is that it has to get first confirmation from a helper T cell. The way that it does that is by taking in that antigen, chopping it up, loading a piece onto an MHC2 molecule so that it shows the whole thing off to the outside. If a helper T cell happens by and agrees that that is indeed a foreign molecule, the helper T cell activates the B cell by making a CD40 ligand, making cytokines that end up activating the B cell. So this is what we've already described. What we're saying now, though, is that there are some instances when B cells can become activated even if there's no helper T cell around to do the confirming. And so here's the situation when that might happen. So B cells can become activated even if it receives no confirmation from a helper T cell, if it happens to be binding to a particular type of antigen. And so a type of antigen that would cause this to happen potentially is a big antigen that's able to bind to multiple B cell receptors, as well as the toll-like receptors on the B cell surface. Right? So when all of these things are being kind of bound by something all at the same time, then that would be sufficient to activate this B cell, even if there's no helper T cell around. So to summarize what we've just shown you here, what if we do this? We'll kind of make these pictures a little bit smaller so we can have room to write some words down. So whenever you're talking about an antigen that is not able to activate that B cell unless there's a helper T cell around to help activate the B cell, that type of antigen is called a T-dependent antigen. And again, it's called that because it's an antigen that will activate the B cell only if there's also an activated helper T cell around. And the thing about T-dependent antigens is that they tend to be smaller and they tend to be made of proteins. On the other hand, we've just mentioned that there's certain types of antigens that can cause a B cell to get activated even if it doesn't receive confirmation from a helper T cell. So whenever you're talking about this type of antigen that can do that, it's called a T-independent antigen. And again, it's called T-independent because it can activate a B cell even without the help of a helper T cell. A couple of things about T-independent antigens. Again, they tend to be relatively big, which means that they're able to bind to several B cell receptors all at once, and they can also bind to any toll-like receptors that are on the surface of a B cell. So they can bind to all of those simultaneously. Another thing about T-independent antigens is that they tend to be made of lipids and carbohydrates. So for example, the capsule. You might remember that bacterial capsules are made mostly of sugars, which is a type of carbohydrate. So capsules tend to be T-independent antigens. And one more thing about T-independent antigens is that they tend to be poor at causing the production of memory cells. So if you remember, we mentioned that one of the things that usually happens whenever a B cell gets activated is that it makes clones of itself. Some of those clones become these really valuable things called memory B cells. What we're saying here is that if it's a T-independent antigen that ends up turning on a B cell, it makes clones of itself, but it doesn't really make memory cells. So those clones don't typically produce memory cells. They tend to produce only plasma cells. And basically the reason why that is because in order for this B cell to know to produce clones that become memory cells, they have to receive cytokine signals that tell it to produce memory cells. And so because in this scenario here, there's no helper T cell around that's spitting cytokines out at it, it never really gets the message to become memory cells. So that's what we mean here by these T-independent antigens are poor at causing the production of memory B cells due to the lack of cytokine involvement. All right, so that's one kind of miscellaneous thing to know about our immune response to exogenous antigen. There are some exogenous antigens that activate B cells even if there's no T cell around to do the confirming. Another miscellaneous thing to note about exogenous antigens is that antigens found deep inside bacteria are able to stimulate an immune response. So that might seem kind of counterintuitive, and so here's what we mean by that. What we're saying is this. So whenever you have a bacterial cell, actually common sense might tell you that the only parts of this bacterial cell that our immune system should be able to see is the outside, right? But what we're saying here is that actually there are molecules that are antigens inside this bacterial cell that can also end up stimulating an immune response. And basically the reason why that is because we have phagocytes like macrophages that can do this. They can encounter that bacterial cell And again, if its toll-like receptor is engaged, that means that it produces a co-stimulatory molecule as a result of that. But whenever a macrophage comes across a bacterial cell, it can take it in and digest it, chop it up to bits. And so as a result of that digestion, now you have molecules that were once buried deep inside that bacterial cell. Now they've just become available. Right? The chopping up causes them to be freed, basically. And once those things that were inside the bacterial cell are freed, they're now able to be loaded onto an MHC2 molecule, for example, and be presented to the outside, where it could then end up activating something like a helper T cell. Right, so when we say that antigens found deep inside bacteria, those can actually also stimulate an immune response. The reason why that is because bacteria that are digested by cells like macrophages can have the things that were once locked inside, the molecules that were once locked inside, now be freed. Right? And so that allows those pieces then to be presented to a helper T cell, which then responds to it. So again, that's basically the story on how we respond to exogenous antigen. And again, you find exogenous antigen on foreign cells like bacteria. What we'll take a look at next is how our immune system responds to endogenous antigen. Remember that endogenous antigens are antigens that are produced by our own cells. And one of the reasons why our own cells might end up producing weird-looking molecules is if they are infected by a virus. And so what we'll take a look at here is how our immune system responds to that type of infection. It turns out that while viruses are still free-floating, so before they've found a cell to infect, it turns out that we have things like B cells and antibodies that might be able to bind to them and neutralize them so that they aren't able to infect a cell. But it might be the case that not every single viral particle ends up getting neutralized, so some of them might be able to slip through the cracks and end up infecting one of our own cells. So whenever that happens, you might be thinking that all hope is lost, because now we have, now our antibodies and our B cells wouldn't be able to get to that virus, so that virus then has kind of free reign to do whatever it wants to do with that cell and make that cell produce more complete viruses. It turns out, though, that we do have a type of white blood cell that's able to take care of this type of infection. So basically we have a cell called cytotoxic T cell that's able to recognize the cells of our own body that happen to be infected with a virus. And when they recognize a cell like that, they can release molecules that end up killing that cell. So the bad news is that cytotoxic T cells actually kill off our own cells, but it does so for the purpose of killing off the viruses that are inside of that cell. So the basic idea of how our immune system responds to endogenous antigens is that, well, here's a major player right here in the response to endogenous antigens. We have cytotoxic T cells that are able to recognize and kill abnormal cells. And by abnormal cells, we're talking about our own cells that have been infected with viruses, or our own cells that have become tumor cells. So then the question becomes, well, how do cytotoxic T cells know the difference, right? How do they tell the difference between an abnormal cell and one of our normal cells, right? So you don't want these cytotoxic T cells just killing everything. You want it to just kill the ones that have become infected with the virus, for example. And so the way that cytotoxic T cells are able to tell the difference between a sick cell and a normal cell is these MHC1 molecules. So that's the key. So remember, we've mentioned before that all of the cells in our body, all of the ones that have a nucleus, that would be everything except red blood cells, they all make these molecules called MHC1 molecules. And basically what MHC1 molecules do is that they continuously sample all of the proteins that are being made inside of that cell, and they show it off to the outside to any cytotoxic T cell that happens to be wandering by. Right, so in words, these MHC1 molecules, they take samples of proteins being made inside of that cell, and they again present it to the outside for a cytotoxic T cell to see. And so here's what that looks like. So again, we have cytotoxic T cells that are continuously kind of patrolling the blood and the lymph. It's just looking for a reason to kill something. Say that you have one of our own cells that just happens to be a normal healthy cell. So if it's a normal healthy cell, all of those normal healthy cells produce proteins that have some normal function for that cell. What we're saying that these MHC1 molecules do is that whatever proteins are being made inside of this cell, these MHC1 molecules are continuously kind of taking a sample of those proteins that are being produced in order to show it off to the outside. And so if it's a normal healthy cell, basically this is this cell's way of saying, hey look, cytotoxic T cell that's in the neighborhood, don't attack me because I'm just a normal cell. Here's my proof that I'm a normal cell. I'm just making normal healthy proteins that you would expect me to make. So if a cytotoxic T cell happens to come across that, it's hopefully its T cell receptor won't bind too tightly to that. So the cytotoxic T cells that are out patrolling the blood and the lymph, they should have T cell receptors on them that aren't able to bind to molecules that you normally find in our cells. And so because it's not able to bind to it, it should disengage and not really respond to it and end up leaving this cell alone. So that's basically the reason why our cytotoxic T cells don't end up harming our normal healthy cells. On the other hand, if you have one of our cells that happens to be infected with a virus, remember that one of the things that viruses do after they infect a cell is that they take over that cell. They hijack it so that it starts producing nothing but viral parts instead of normal cell parts. So if you're this cell, now you sense that you're in trouble. You've just gotten hijacked, and it's in your best interest now to flag down a cytotoxic T cell to come basically kill yourself so that the viruses that are inside of it don't end up spreading to other cells of the body. And so here's how this infected cell calls for help. And so the key again are these MHC1 molecules. And again, all these molecules do is that they take a sampling of whatever proteins are being made inside of the cell and they show it off to the outside. So if it's a cell that's infected with a virus, this cell is now no longer making normal proteins, it's now making viral proteins. And so this is what's going to end up being sampled or loaded onto this cell's MHC1 molecule that ends up getting then showed off to the outside. And so basically what this cell is saying is, look, I'm in trouble. I've just gotten hijacked. I need some help from a cytotoxic T cell. This is my proof that I've been hijacked. So please come help me out. And so if a cytotoxic T cell happens to run across that, again, the cytotoxic T cells that are patrolling our blood and our lymph, they should all have T cell receptors on them that are able to recognize foreign molecules. And so actually, in order for this cytotoxic T cell to become activated, it not only has to see an antigen that's being shown to it, but it also has to have cytokines that are being secreted at it by a helper T cell that itself became activated by, say, a dendritic cell. So once this cytotoxic T cell kind of sees those two things, an antigen that's being shown to it and cytokines that's being secreted by a helper T cell, that's what causes the cytotoxic T cell to become activated. And when we say that it becomes activated, what we mean is that it ends up making clones of itself, it ends up copying itself. Some of those clones become these things called memory cytotoxic T cells, whereas other clones become these things called effector cytotoxic T cells. And again, remember, whenever you see this term effector as part of a cell's name, it just means that this is a cell that's ready to perform its function. And here's what the function of a cytotoxic T cell is. So when an effector cytotoxic T cell happens to run across one of our own cells that's showing it danger, that's showing it that it's been hijacked by a virus, this effector T cell then ends up killing this cell. And so maybe we'll point out here that this molecule that you see on this cytotoxic T cell, remember that helper T cells have a molecule that's very similar to this called CD4. And the function of this molecule was that it helps to stabilize the interaction between the MHC, the antigen, and the T cell receptor. Cytotoxic T cells, their molecule that does that stabilizing, it's not a CD4 molecule, it's a slightly different molecule called a CD8 molecule. So in other words, one of the ways that someone could identify a cytotoxic T cell is that it has CD8 molecules on them. This is different from helper T cells, which have CD4 molecules on them. But that was kind of an aside. But so whenever this effector cytotoxic T cell now sees a cell that's showing it danger, it ends up killing that cell. And the way that it kills it is by releasing these molecules called perforins and proteases. And so you might guess from the name that something called a perforin, what that does is that it causes perforations or holes in this cell's membranes. After those holes have been produced, then these enzymes called proteases can actually rush inside of the cell and cause the cell to die. And actually, cytotoxic T cells are smart. So the way that they cause this cell to die, it doesn't cause it to die a messy death. Right? So if you're a cytotoxic T cell, probably the last thing you'd want to do is cause the lysis of this cell, because if this cell bursts open and if it dies that way, then all of the viruses that are inside of it then can escape and spread to other cells. So that's kind of a dumb way to end up killing this cell that's infected with the virus. So instead, what the cytotoxic T cell does is that it causes this cell to die by undergoing apoptosis. So it basically ends up shriveling up so that a macrophage can come eat it, and when the macrophage takes it in, it digests it. So that if there were any live viruses inside of that cell, it's now just become pulverized. So then, to sum this up, we're taking a look at how our immune system responds to endogenous antigen. These cytotoxic T cells play a really big role in responding to endogenous antigen. Specifically, we have these effector cytotoxic T cells that cause apoptosis of abnormal cells. After that cell that's infected with the virus undergoes apoptosis, it then is ingested by a macrophage, and that's how it ends up dying. One interesting thing that has ended up happening is that there are some viruses that have actually evolved the ability to prevent being killed by cytotoxic T cells, and so here's how they do that. So there's some cytotoxic T cells there are some viruses that have acquired the ability to do this. Once they have gained access to a cell and have infected that cell, they actually have ways of preventing that cell from producing these MHC1 molecules. And so from the virus's point of view, that's really good. That's really clever, because now the virus can hijack this cell and cause it to produce nothing but viral proteins. And because this cell now is lacking MHC1 molecules, it has no way of flagging down a cytotoxic T cell to come kill it. Right? So if a cytotoxic T cell does happen to be in the neighborhood, it has no way of knowing that this cell is in trouble, because again, the virus has stopped this cell from making MHC1 molecules that would flag down the cytotoxic T cell. So again, it's pretty clever. So we're saying here that some viruses can avoid being killed by cytotoxic T cells. The way they do that is by downregulating the expression of those MHC1 molecules in the host cell. So again, that's really clever from the virus's point of view, and it's really diabolical, and it might worry you. So you might be thinking, well, oh shoot, what if I happen to be infected with one of these viruses? Does this mean that I'm going to die? That I have nothing to take care of those types of viruses? Well, kind of another interesting thing on top of this interesting thing is that we've actually evolved the ability to fight back and deal with these types of viruses, and the way that we do that is by way of these natural killer cells. So we've already seen that natural killer cells participate in ADCC, in that they can kill bacteria that have been coated with antibodies. So we've already seen that. It turns out that that's not the only thing that natural killer cells can kill. Another thing that they can kill are our own cells that happen to have been infected with these clever viruses. So these natural killer cells kind of can recognize which of our own cells have been infected with these clever viruses and release perforins and proteases to kill it off. And basically the reason why this works is because MHC1 molecules actually turn off natural killer cells, right? So here's what that looks like. So if you have one of our cells that's just a normal healthy cell, And if it's a normal healthy cell, it's just making normal healthy proteins, and it's also able to produce these MHC1 molecules that show off some of these normal proteins to the outside. So these normal healthy cells, they're not attacked by cytotoxic T cells, right? Because cytotoxic T cells will see this and say, oh, that's just a normal protein, I know not to attack you. The reason why a natural killer cell would not attack this normal healthy cell is because it turns out that this MHC1 molecule itself happens to deactivate, bzz, bzz, gives an off signal to natural killer cells so that it ends up not attacking that cell. Right, so then back to this point, so the reason why the natural killer cells can kill viruses that prevent the expression of MHC1 is because if this natural killer cell runs across this cell that has no MHC1 on its surface, then that means that there's nothing to turn off this natural killer cell. And so that's how this natural killer cell knows then to kill this cell that's been infected with a clever virus. So to sum this up, we're saying that natural killer cells, they can actually kill cells that lack an MHC1 molecule. And basically the reason why that works is because these MHC1 molecules, they actually inhibit the activity of natural killer cells. So MHC1 molecules, they turn off natural killer cells. So that's the overview of how our immune system responds to endogenous antigens. There's basically two players involved. There's cytotoxic T cells and natural killer cells.